The overall goal of the following experiment is to gain quantitative structural information about E. coli cell division proteins in their native environment. This is achieved by expressing a photoactivatable fluorescent fusion protein in live cells and immobilizing these cells on an agarose gel pad. The cells are then imaged via streaming video, while excitation and activation lasers are applied to visualize single molecule locations. The resulting movies are computationally analyzed to extract the molecule positions with nanometer accuracy and superimpose them to generate a super-resolution image. This image can then be analyzed by a variety of quantitative measurements that describe the structure of interest, including its dimensions and molecule density.
Photoactivated localization microscopy, or PALM, is an easily implemented super-resolution fluorescence microscopy technique. The instrumentation is straightforward, but careful optimization of data acquisition and analysis is required to generate reliable results.
Another crucial step is the extensive characterization of your fusion protein and expression level to ensure that your results are biologically relevant. The following protocol was optimized for the FtsZ protein fused to MES2 and should be adapted to your protein of interest.
To begin, inoculate LB media with a single colony of the strain of interest containing a fluorescently tagged protein prepared as described in the written protocol accompanying this video. Grow overnight in a shaker at 37 degrees Celsius. The following day, dilute the culture 1 to 1000 into M9+ minimal media. Grow to mid-log phase in the presence of appropriate antibiotic at room temperature, and then induce the culture to produce protein with 20 micromolar IPTG for 2 hours. Meanwhile, assemble the imaging chamber by making a 3% agarose solution in M9- media. Melt the agarose at